and I will share my screen here. Okay. 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 Can you see my screen? So this is the
Yes.
top
Yes,
of
sir.
the
Yes, sir. Yes,
Yes,
sir.
slide
sir.
and the bottom of the slide. Can you see the
Yes,
pointer?
sir.
Yes, sir.
Okay. So last time when we started kind of talking about DNA sequencing, uh, I know it was like stop, start, stop, start. We kept going in and out. So what I'm going to do is just so that everything is clear, I'm going to just restart DNA sequencing, kind of cover a little bit of what we did last time, and then take it on from there. Okay. So DNA sequencing is one other aspect of biotechnology which one needs to know, because one will be using it for several reasons. Okay, and as far as nowadays is concerned, DNA sequencing is part and parcel of many areas of expertise like mall bio, cell bio, biology, developmental biology, genetics, pharmaceutical chemistry, pharmaceutical biotechnology, zoology, botany, etc., etc. Okay, and it is good to know how one can do sequencing of DNA just as a part of your general knowledge. Okay, it's something which we should all know. Okay, but what do we mean by DNA sequencing? When we say that we are doing DNA sequencing, it is what we are trying to do is determine the order of nucleotides as they occur along the DNA strand. So if you remember, the DNA strand has got a sugar phosphate backbone and extends from what we call as a 5' phosphate all the way to the 3' OH. Now, on each of the sugar phosphates, there is a base. And this sequence, as we look at the DNA from the 5' to the 3' end, is referred to as a DNA sequence. Okay? Now, the reason one likes to know what is the sequence of DNA is because on one hand, every organism, what we call as every living organism here, has got DNA as its genetic material. Okay, and all of its functions, whether it is life functions or replicative functions, are all encoded in this DNA, which we call as a blueprint of life. So knowing the sequence of DNA of a particular organism gives you a lot of information about how that organism might function, okay, and what are the properties of the organism, okay, and therefore, as I say, DNA sequencing is fundamental to analysis of the genome and understanding biological processes in general and biological systems in general. Okay, now DNA sequencing was actually uh, invented, I should say, in 1977 by two people, almost like just about a few months apart. One was Sanger, Frederick Sanger, and the other was Gilbert. Okay. Uh, Now both the people didn't do it alone. Sanger did it with two other people. I think one of them was Coulson and other one, I'm trying to recall the name. And Gilbert did it along with his graduate student Maxim. Okay. Now, even though discovered in 1977, Sanger method is still used even nowadays. Okay, in many laboratories, it's very accurate, and it is used many times as a standard against which newer methods are compared. Gilbert method, which is also very accurate, simply because it uses a lot of toxic chemicals, has now gone out of favor. Okay, and very few labs probably use it as of today. Many of the labs nowadays use this more advanced sequencing, what we call a cycle sequencing, or next generation sequencing like 454, pyro sequencing, etc. Okay. Now, DNA sequencing has not been easy. Okay, as of today, it might appear to be easy, but at one time, it was a fairly painstaking process, and which took a lot of effort. And because of that, the cost of sequencing DNA was also very high. So, in and around, like say 2000, this is a time when human genome was being sequenced. The cost of sequences was close to about 100 million for a genome. Okay. But see what has happened over the many years. As we are approaching, 2014 here, the cost has gone to even less than 10,000. As of today, the cost of sequencing a genome is roughly between 1,000 to $2,000. Okay, so imagine the big, decrease in the cost as you go along, and part of it is because of this new sequencing techniques called as next generation sequencing techniques. Okay, I might maybe, if time permits, talk just a little bit about this next generation sequencing. But much of what we are going to talk about as far as the syllabus is concerned is the older, more conventional methods of sequencing called as Sanger chain termination sequencing, or also called as Sanger kind of biological or enzyme mediated sequencing. And the second method, which is called as the Maxam and Gilbert method, or is also referred to as chemical sequencing or chemical induced degradation and sequencing. Okay. Now, many of the advances in sequencing, which include automation and robotics, have been applied to both of these methods. Okay, so what was very time consuming when they were first discovered, later on became fairly routine because they were amenable to robotics and automated sequencing. Okay, so let us begin a discussion with Maxam and Gilbert sequencing. As I referred to you, Maxam Gilbert sequencing was developed by Alan Maxam, who was a graduate student or a PhD student in the laboratory of Walter Gilbert way back in 1976-77. Okay, and the basic principle, as I indicated even last time, is the nucleobase specific partial chemical modification. Okay, which means we are going to very specifically modify the base which is present on the sugar, whether it's adenine, guanine, thymine, or cytosine. And after this modification is done, it is followed by the increased lability of the DNA, and therefore the DNA is cleaved just at sites adjacent to where this modification has occurred. Okay. Now, I just changed the presentation from last time because I got some time in between to make it a little more interesting. So this is the paper which was published indicating that they had invented a new method for DNA sequencing by Maxam and what do you call as Walter Gilbert. It was in the Proceedings of the National Academy of Sciences, a very prestigious journal, in February 1707. Okay, this is Walter Gilbert here. I tried to find the picture of Alan Maxam, but for some reason, being a student there, he never got the fame that his guide got. So I wasn't able to find the picture. Maybe if I find one day, I'll give it to you or show it to you. Okay. So this is historically, a classic paper. If anybody wants this paper, let me know. I have got a copy of this, and you can read just for personal interest. How these techniques first were presented. Okay. Now, the steps in Maxam Gilbert sequencing are summarized here. Okay, so they include about 7 steps, beginning with what we call as labeling the 5' end of the DNA that is to be sequenced, and then distribution of the DNA samples into different sample tubes, which will be subject to different chemical reactions, and these chemical reactions will result in what is called as base modification. Okay, and after this is done, you put another reagent to do cleavage adjacent to where the bases have been modified. And then these broken pieces of labeled DNA are separated based on size on gel electrophoresis, and then looking at the size and where the bands appear in terms of the origin in which of the 4 tubes, you can then detect where these samples are by radiography. And looking at that, you will get to know what is the sequence. Okay. Rather than worrying about what this means, as you go through each and every step, the reason for these terminologies will be apparent. Okay. So let us begin our discussion. Let us say, just for argument, this is a piece of DNA whose sequence I want to find out, and the sequence is unknown at this moment. So when I want to do Maxim Gilbert sequencing, I can use either one of the strands, okay, either this strand or this strand to know the sequence, okay. And Maxim Gilbert method is only applied to single strand. It is not applied to double-stranded DNA. Okay, but once I know the sequence of one strand, because of the rules of Watson-Crick base pairing, by default I do know the sequence of the other DNA. Okay, so for example, if I know this sequence is AT, GC, CG, I know A base pairs with T, T base pairs with A. So by default, I also know the other sequence. But if I want to know the unknown DNA sequence, all I need to do is find the sequence of either this strand or this strand, and by default, I know the other one. So in the first step, what is done is that DNA, that is double-stranded DNA, whose sequence I want to find out, is separated into two single strands. And one can do it either by increasing the temperature, and if you know, remember, when I increase the temperature, the DNA will melt and it will become single stranded, or I can add NaOH or change the pH or the ionic strength, causing this hydrogen bonds to break down, giving me two single strands. Okay. Now, after this is done, what is typically done is the mixture of the two single strands that you have is run on a gel. Okay, and when you run on the gel, you will find that invariably one strand will move differently than the other strand, and the reason is because in either strand you will find that the actual content of purine to pyrimidine will vary. Okay, just for example, look at this strand. This is a purine, this is a purine, this is a purine, and this is a purine. So there are four purines, A, G, G, and A. And see the remaining 1, 2, 3, 4, 5 are pyrimidines. Okay. Now this is an example of a very small piece which I have shown for example. But many times when you do DNA, you want to sequence 100 base pairs, 200 base pairs, 500 base pairs. In that case, you will find that one of the two strands is always richer in purine, while the other one is always richer in pyrimidine. Okay, because they are purine pyrimidine base pairs. So when you separate the two strands and you run it on a gel, you'll always find one moves more than the other. Okay, so this is what we call as the, in a sense, the lighter strand. Okay, lighter will move further than the heavy one, and then this one is the heavier strand. Okay, now you can take a sample from either of these locations. Okay, and for your purposes, you can use that for your sequencing. Okay. Now, let us presume that I am taking this band here. Okay. So this is the one which has got, according to this argument, more purines. Okay, whatever it is. Okay, but whichever strand I take, what I then do is first remove the phosphate which is already present at the 5' end by treating it with alkaline phosphatase, and after that I treat it with polynucleotide kinase in the presence of radioactive ATP. So when I do that, this kinase will add a new phosphate group, but this phosphate group, because it is derived from radioactive ATP, will contain P32, which will make this DNA now labeled. Okay, in other words, I can track this DNA by the fact that it will show radioactivity. Okay, so the only reason I label is so that I can follow the DNA through all the procedures that I'm going to do later. Okay, now as I said, you can take this strand also and do the same thing. The difference will be that that strand will have the opposite orientation and the band will appear to be here relative to this strand. Okay, or the phosphorus will be here relative to this strand. Now once this is done, then you go to the next step. What you do is take the DNA and actually distribute it into 4 tubes, okay, and distribute it equally. And in tube number 1, what you do is you add this reagent called as dimethyl sulfate, okay, dimethyl sulfate. In tube 2, you add dimethyl sulfate plus formic acid, okay. Now, for example, in the next slide, I'm going to show what happens when you add DMS. Okay, so when you add DMS, what will happen is, if this is the strand of DNA that you are having, and here is the label here at the 5' end. Okay, see this? This is phosphate here, which is shown as a little wedge. Okay. Now, when I treat with DMS, what will happen is, randomly at any one of the guanines here, I'll I'll get methylation. Okay, so DMS actually does methylation. Now note in this strand that there is another guanine here. Okay, so as an example here, what I might find is, in this piece of DNA, either there will be methylation here, or there will be methylation here, or there will be methylation at both the places. Okay, and what I do is, I make sure those probabilities can occur by keeping DMS in limiting concentrations. Okay, if I add too much DMS, then every guanine which is present on this DNA, all of them will get methylated, and I do not want that to occur. And I'll tell you why I don't want that to occur. Okay, so now let us see. Yeah, hello.
Sir, well, limited quantity, how can we predict what groups will be methylated? Isn't that
Correct.
random?
It is very random. Okay, I actually don't predict. I want it to be random. I want that in some of the DNA strands, methylation occurs here. I also want, by random event, in some of the DNA strand, methylation to occur here. And a random event could also be methylation occurring at both places. Okay. But see what happens when methylation occurs here. When methylation occurs here, and remember, in the presence of DMS, after methylation occurs, you actually get deep urination. And when deep urination occurs, at this point there is only ribose. Okay? And later on, when you treat with this reagent called piperidine, which is for cleaving the sugar phosphate backbone, what happens is the sugar phosphate backbone gets cleaved here. Okay? So now, basically, I'm going to get 2 pieces. Okay? Now see how the 2 pieces are. This piece will contain this C and A, which was on the 5' end of the guanine. Okay? And this piece will contain all the other bases and sugar phosphate, which was downwards from this guanine. That is on the 3' side. But in terms of detection, the only piece that I can detect is this one, because this is the only one which carries the radioactivity. Okay. Now, imagine for a moment that in at another molecule, this guanine was methylated. If that were the case, what would have happened? I would have seen a methyl group here. Then it would have been deep urinated here. Then it would have been cut here. And when it got cut here, what I would have found is a piece which was C, A, G, and T, which is still radioactive. And these 2 things would have disappeared and not be traceable. Okay. And now you understand why I need limiting quantities, because I want to generate all the possible pieces where the cut has occurred where there was a guanine, like this piece and this piece. Okay. Likewise, when I add formic acid also, then what happens is, there is a chance that this gets methylated, and if this gets methylated, it will get cut here. There is a chance that this gets methylated. If it does, then it will get cut here, and I'll have a piece which is Ca. Then it's possible that this gets methylated and cut here. Then I have a piece with 4 bases. Or it gets cut here, then I have a piece with 5 bases. So basically, I'm going to generate all possible pieces because of cut, either at adenine or guanine when I add DMS and formic acid. Now I do the same thing in test tube number 3 and 4, where I had either hydrazine or hydrazine in the presence of sodium chloride. Just like I showed before, if I, let us say I had added hydrazine, then I would have got a cut here. And if I got a cut here, what would have happened? I would have just lost the phosphate, and that was it. I would not detect anything. But I would have also got a cut here in hydrazine plus sodium chloride. That would be 3 long. It will be 1, 2, and 3, and so on and so forth. Okay, and cut here. Okay, so basically what I'm trying to generate is small pieces of DNA at every location, whether they're either a cytosine or any of the two piperidine, that is cytosine or thymidine. Now, you have to realize now that in the 4 test tubes, where I've added in one case DMS, in the other case DMS plus formic acid, in the third case hydrazine, and in the fourth case hydrazine plus sodium chloride, I am going to, because of random reactions, generate pieces of different lengths. Okay. Now remember one thing, if I add too much reagent and I wait for too long, then what will happen is, in the tube, for example, if let us say this is a sequence, in the tube where I've added DMS, okay, and I waited for long time so that every guanine got methylated, then the only piece I'll generate will be 80, because all the other pieces, even if they are formed, will not matter, because as soon as this cut is occur, further cutting here will not be traceable because there is no phosphorus. Similarly, when I treat with DMS plus formic acid, what I'll get is, I'll get a cut here, okay, and all these pieces will be lost, and I'll see nothing in that test tube when I do the next step, which is electrophoresis. Okay, so I have to add reagents in quantity such that all the pieces are generated, and then what I do is do a gel electrophoresis. Okay, now last time I had shown you some reactions. In this slide I showed more simply, okay, where adenine in the presence of DMS gets methylated here on what we call as carbon number 1, nitrogen, which is numbered 1, and guanine gets methylated at this nitrogen, which is N7. Okay. Similarly, when I treat with hydrazine, whether it is cytosine or thymine, what happens is this ring here gets actually rearranged to a 5-membered ring, and then later on when you treat with what you call as piperidine, you will lose the base and you will cause a cut where that base was. Okay. Now these are the slides which show all the mechanisms, probably not required, but I've included in this presentation. Later I'm going to give you this PPT, okay, so don't worry. Okay. Now here is what happens when you do Maxim Gilbert sequencing. So again, I'm showing with an example here. Okay, let us say this is a piece whose sequence I'm trying to find out. Okay, and I've labeled it with P32. Okay, now this is the tube where I've added, if you remember, DMS plus formic acid. This is a tube where I've added DMS. So the cut is occurring wherever there is a G. Here it is occurring wherever there is a ANG. This is hydrazine, hydrazine, and this is hydrazine plus sodium chloride. Okay, remember, hydrazine plus sodium chloride cuts only at C, and hydrazine cuts at C plus T. Okay, so this is hydrazine plus sodium chloride, and this is hydrazine. Okay, now let us look at it simply. Let us look at this one first. Okay, so this is where I've added only DMS. So see where the cut is going to occur. The cut is going to occur where this G was originally. When that happens, I will simply lose this phosphate. I'll generate this strand which I will not be able to detect because the label is lost. And where is the other cut going to occur? Between the C and G. When it occurs between C and G, I'll generate this piece, which is GCTAC. GCTAC, which will have a phosphate tag. Now what happens when I add DMS plus formic acid? So again, a cut will occur here, but that will result in loss of phosphorus. I will not be able to detect this piece. Then the cut will occur between this T and A. Okay, and that will give me a piece which is GC and T. Then a cut will occur between C and G. That will give me a piece which is GCTA, just like this tube. and I'll get another piece where the cut occurs between T and A, which will be GC, TA, CGT. GC, TA, CGT. Okay. Now look at this test tube where I've treated with hydrazine plus sodium chloride. It cuts between G and C here. So I get this phosphorus tag and guanine here. And it also cuts between A and C. So I get GCTA. Okay. Now look at this one. I generate these two pieces here. Plus in addition, because it cuts after T, I generate a GC piece and I generate a GCTACG piece. Okay. Now if you look very carefully, the length of pieces in the purine cutters tubes and the length of pieces in the pyrimidine cutting tube will never be the same. Okay. So there will be no match. Okay, in the two tubes, they will be mutually exclusive. So if you see, piece of one length, which is G, is present here. Piece of two length, which is GC, is present here. But 1 and 2 are not present here. Here is present the length 3, which is not present here. Here I see length of 4. Okay, 4, which is present here and here. Then 5 is present here. Then 6 is present here. And 7 is present here. So the pieces that are generated at random will be such that all the sample tubes which are cut with DMS or DMS plus formic acid will have pieces whose lengths are always different from those tubes in which I have added hydrazine or hydrazine and sodium chloride. Now when I do the gel electrophoresis and I separate the pieces on size, the smallest piece will move furthest. Okay, so this G1 will be here. And remember that piece is also there in this test tube. So I'll see a band here when I expose them to radio to X-ray film. Okay. And the P32, remember, will expose the film and I'll see a radioactive signal. Now see this piece GC will appear in this sample tube, but will not be here. Then this will have the 3 piece. Okay. GCT. Then the 4 length is present both in this tube and this tube. See this? Then length 5 is present in both these tubes. Okay, here length 6 is present in here, this tube, and length 7 is present here in this tube. Now remember, when a band appears in both the pyrimidine containing tubes, it has to be C, because this is C and this is C plus T. And a band which appears only here will have to be T. Okay, that is cut at T. So all I have to do is read the sequence from here up and I'll get the sequence of my DNA. So see the sequence, I'm reading C, T, A, C, G, T and A. And look at it, C, T, A, C, G, T and A. Now, if you are smart, you would have realized that one of the drawbacks of the Maxim-Gilbert method is that I can never know what was the first base. Okay. See, if you look very carefully, the first reading I'm getting is only after it cuts here between G and C. The time it cuts before G, I simply lose the phosphate. So that they say is one of the problems with the Maxim-Gilbert technique. The only base you cannot find out is the first one. All the others you can easily find out. Okay?
So,
Is that clear? Yeah.
so like in C and C plus C, the common base is G and the common segment is G and P. G with the radioactive P, right? So, like when we
No.
do a gel so when we do the gel electrophoresis of C, so you will be having this G with the radioactive P going more further as compared to
Yeah.
that down.
Here.
So why? So
So
why?
see, for example, if I look at G, no, it has got 5 bases, right? So it will move here. But look at this, which has got 3, 5 and 7. So this is 3, this is 5 and this is 7. Similarly here C, shortest moves here. Okay. And here C plus T also, that is there, that moves here. Okay. So again, what was your question?
My question was कि then when we are starting from C, like we are giving the name to the segments, right? Like we are counting ki C and C plus C. So if it is present in both, then we are giving it a name of C.
Correct.
But then it's a G, right? मतलब the fragment in this is G,
No,
right? So
no, no, no. What we are saying is the fragment ka end में there is a G, but where did it cut? It cut where there was a C, no? You see this? See, because I treated with hydrazine and hydrazine modifies cytosine and hydrazine plus sodium chloride also modifies cytosine. So what I'm saying is the piece I'm getting here is a piece which was modified at the C and cut where the C was. And when it was cut where the C was, I was left with what was G. Okay? Are you seeing what I'm saying?
yeah, got it, sir.
It is not telling me what is going to be at the end. It is telling me where the cut occurred. So here the cut occurred at C. So I got these two pieces. Then here the cut occurred at T, right? So I got this piece which contains GC. But I really don't know what this is. Only I know is there's a radioactive piece here. And this radioactive piece, whether it contains G or not, I do not know. Okay, are you seeing? I only know there's a radioactivity. Okay, so that's
Yes,
why
sir.
I cannot find out what is that first base. Okay, so I start the sequence from the second, third, fourth,
So,
fifth, sixth, seventh, eighth. Yeah.
so what would be its feasibility if we scale up? Like, uh, if the DNA we want to sequence is like having a million base pairs, so it would
Mm-hmm.
be like a hello.
Yeah, yeah. Go ahead.
So it would be a tedious process to know like which by this method. So, sir,
Exactly. You are right. You are very correct. Maxim Gilbert method works best when you have DNA sequences which are only up to 100, 150 to a maximum of around 500. Okay. Anything more than that, it creates a problem. Okay. So when I want to sequence huge lengths of DNA, which are like E. coli DNA, which is million bases, then I have to first cut the DNA into shorter pieces, which are manageable, and then what I do is sequence them, and then after sequencing them, I put the sequence together. Okay. If you remember when we constructed genomic library, we used to cut into small pieces which are overlapping. Now imagine I take each of those clones and I cut open the plasmid to give you one piece of DNA. I sequence that. Because of equal overlapping sequences, I can put together the entire genome. Okay, so you are very correct. You cannot do Maxim Gilbert sequencing on huge pieces of DNA. The DNA pieces have to be manageable. Generally speaking, we do about 100 to 150 to 200. Okay, but if you're really, really good and you've got good hands, good experience, you can get very accurate results up to 450 to 500 bases. Okay, so does that answer your question? Hello?
yes, sir. Yes, sir.
Yeah.
Sir, also, is there any machine or technical instrument that counts this base pair? So it has to be done manually.
Okay, in this case, you do it manually. When you do auto radiography, no, uh, you almost have to do it manually. Okay, the instrument can probably give you a nice picture of good quality, but other techniques, which I'm going to show you a little later, the Sanger method, there's a machine which will do all these things for you, and I'll show you how it does it. Okay, so you don't have to physically do this, uh, observe and then do. Okay, but in the olden days when we did Maxim Gilbert, we had to physically do this. Okay, expose it to radioactivity, make sure the paper doesn't, X-ray film doesn't move, and then after you get that film exposed, you have to physically do this thing, just like I did it now. Okay, like say C, T, A, C, G, T, A. And usually one other person will sit with you so that you don't make a mistake. So you read out and you verify kind of thing. Okay, but nowadays we don't do that. There are machines, and I'll show you how it is done. Okay. So the features of Maxim Gilbert is that it's a direct method and you get a direct readout, just like I showed you. It's very accurate and it's very sensitive if done correctly. Okay. But the drawbacks are we can never know what is the first nucleotide. And again, as I said, answer to your question, it cannot read more than 500 base pairs. And also it takes some experience to set up those reactions so that you get all the pieces. Remember I told you, you want a reaction which doesn't go all the way and does methylation everywhere so that you generate only one type of piece. But you want to generate all the different pieces. But more important, the chemicals that are used, hydrazine as well as dimethyl sulfate, are very toxic. And not only that, because you are labeling with P32, you tend to have to work with radioactivity, and that sometimes is risky. Okay, you can get yourself exposed to radioactivity, and you have to be very, very careful. Okay. Now, as I said, at about the same time, in another lab, another technique was invented, which was called as DNA sequencing with chain terminating inhibitors. Okay, so as I said, there were three people, Sanger, Nicklin, and Coulson. Okay, and see, this was also published in Proceedings of National Academy of Sciences, but in December. Anyone remember which month the previous paper was published? The Maxim Gilbert. Anyone pay attention? Okay, so here we go.
1977.
77, but see, February. That was in February, okay, and this was in
Yes, sir.
December, okay. So about 10 months
Yes.
later, this fellow, and this is Frederick Sanger, okay. Now, what is the basis for the Sanger, as they call, dideoxy method? Okay, in this method, which is what we call as enzyme-mediated sequencing, we use a very unusual nucleotide, what we call as a dideoxynucleotide. Normally, in DNA, if you remember, the nucleotides which make up the DNA chain are 2' deoxynucleotides, where the OH which is present at the 2' position has been replaced by a hydrogen. Okay, and here at this position is typically A, T, G, or C. Now, for the Sanger method to work, what we use is a base which is a dideoxy base. Dideoxy where it is a 2', 3' dideoxy. That means this OH has also been removed and replaced by an H. Okay. So if this is a deoxy NTP, this is dideoxy NTP. If this were adenine, this would be deoxy ATP. If this were, as I say, adenine plus no OH here, adenine and no OH here, we call it dideoxy ATP. Actually, this structure is not ATP. This is got OH here, but imagine there are 3 phosphates, just for argument. Okay. Now, see what happens when you try to do DNA replication when both are present. Okay, in the same test tube or in the same container. Okay. Now, if let us say this is a growing strand and this is a template. Okay, remember I read the template and then I add the incoming nucleotide. So let us say there is a T. So the DNA polymerase brings a A. Okay, then it goes along the strand and it sees a C. It brings a G, and then it makes this phosphodiester bond. Thereby you get this polymerization reaction. Then what the polymerase will do is go further and it sees a G, then it will bring a CTP, and then this reaction will occur and the chain will start growing like this. So what the DNA polymerase does is very systematically read this and then only allow the correct base to come here. Now imagine you are doing this reaction where along with your regular GTP, there is also dideoxy GTP. Now after this reaction occurs where GTP should normally come, if by chance dideoxy GTP came, then I will form this phosphodiester bond, but later on when the DNA polymerase sees a G and it brings a C here, then I'm unable to polymerize because there is no OH. Because unless that OH is there, I cannot grow the chain, because OH attacks this phosphorus, releases the pyrophosphate, and that's how I get polymerization. Okay. So this unique feature of dideoxy nucleotides, in that they, if they are part of the DNA chain, will not allow the chain to grow further, is the basis for what we call as the Sanger's chain termination sequencing. Okay. Now see what you do in reality. Okay. In reality, what you do is get enough DNA, or if you don't have enough DNA, you do PCR and get your DNA. Then what you do is take your DNA and put it into 4 tubes. Okay. And here it says prepare a PCR reaction mix. Actually, it's a polymerization reaction mix. So typically what you will do is that in this reaction mixture, okay, depending on which DNA you want to sequence, you will add a primer, you will add polymerase, then you present a template, okay, and this template is the one whose sequence you kind of trying to figure out the sequence of. Okay, so single-stranded DNA template, this is the sequence which is unknown. And what do? Is you add all the 4 deoxy NTPs. Okay, what do you mean when I say all 4 deoxy NTP? I add deoxy ATP, deoxy GTP, deoxy CTP, deoxy TTP, and this is the interesting part. And depending on the 4 test tubes, in each test tube you either add one of these. Okay, so in one test tube I'll add DD ATP. In the other one I'll add DD GTP. In the third I'll add only DD CTP. And the fourth I'll add only DD TTP, and then I run the polymerization. And after I do that, I do a gel electrophoresis. Okay. But see, in practicality, what is going to happen? Okay. Now let us say this is the sequence I want to know the. This is the DNA I want to know the sequence of. Okay. In blue is the unknown sequence. Okay. What is the unknown sequence? It is CT, GA, CTT, CG. Okay. What I do, and for the moment, do not worry about how this is done. What I do is, to this unknown sequence, I add a known sequence which is known to me. In this case, it is ACAA, and I ligate it. Okay, I just join it together. Okay, so now I have a piece of DNA which has got the unknown sequence, which I am trying to find out the sequence of, and a little piece which is known to me. And this known piece I use to design a primer. And see the primer I make. The primer I make in the 5' to 3' direction is T, T, G, and T. Okay. And what I do is I take this piece of DNA which I want to sequence, I put it in all the 4 test tubes. After I put it in all the 4 test tubes, I add this primer. What primer? T, T, G, and T. I put that primer in all of them. So in all the test tubes, I'll find that the primer will come here and sit like this. Okay. And now when I add DNA polymerase and all the deoxynucleotides, then the polymerase will be able to add the correct complementary ways in the 5' to 3' direction. So essentially, if everything goes well, it will add a C, G, A, G, T, C, A, and G. Okay, but see what I have done in the 4 test tube. In this test tube, I have also put DD ATP or dideoxy ATP. In this, I have put dideoxy TTP. In this, I put dideoxy GTP. In this, I put dideoxy CTP. So see what will happen here. Whenever there is a need for ATP, the chances are either ATP will go correctly or dideoxy ATP will go, because the DNA polymerase is unable to actually distinguish between ATP and dideoxy ATP. So for example, when here I need to add an A or here I need to add an A, I could add either deoxy ATP or dideoxy ATP. Okay, and the consequences of that are different. Okay, now see what happens as an example. Okay, so this is a test tube which contains dideoxy CTP. Okay, so this is what is happening in test tube number 4. Okay, so look at this. So this is the sequence. Primer comes and sits here, and it sees a G. The polymerase sees a G, so it says, "Oh shit, I have to add a C," and it does something very interesting. it either adds a regular C. If it adds a regular C, what will happen? The C will be added, the reaction will continue, and then G will be added, A will be added, etc., till I reach the next G where I have to add a C. But if I by mistake add dideoxy CTP, then what will happen is, for this chain here, as soon as this addition occurs, the chain will not go any further. And I'll generate a piece which is only about 5 more long, which is CT, GT, C. Now, what happens when I come to this point? When I come to this point, if I add a regular C, then that will be added, and I'll further add A and G. But at this point, if I by mistake put dideoxy CTP, then as soon as that is added, my chain will not go any further. Okay. So, what will happen in this test tube is that I will get the complementary sequence all the way, where I've added 1, 2, 3, 4, 5, 6, 7, 8, and 9 bases. But in addition to that, I might also get a small piece which is only 5 long, and another piece which is 5, 6, 7, 8, 9, 10, 11 long, because at those places by mistake I added a dideoxy CTP. Now, such things will happen in all these different test tubes, but at different places. Okay, just for argument, in the tube which contains DDATP, this issue will occur wherever I need to add a A either here or here or here. So I'll get 3 different size of pieces. One where I added A, dideoxy A here and it never extended. Second, I added correct A or dideoxy A here where it stopped. And then third, where I added correct A here and here, but dideoxy A here, and then I get a longer piece. Okay. Now, essentially, what you are going to have, what is going to happen is, wherever you have added the appropriate dideoxy CTP, there is where the reaction is potentially going to stop. Okay. So, now, when you add polymerase, as they say, the primer will be extended till DD-NTP is added. Okay. As soon as the DD-NTP is added, the chain will end. And what you essentially have to do is, in the test tubes, just maintain the correct ratio of your regular triphosphate, nucleotide triphosphate, and dideoxy, so that the termination will occur at different places throughout the length of the template. And because of that, what will happen is, you will get pieces of different sizes. Now, what you do is, you take those pieces and do a gel electrophoresis. And because of the different size of pieces that you will get, you will be able to now do the sequencing. Okay. And I'm going to show here, let us take this example here. It's very nice here. Okay, so this is the sequence whose whose sequence is unknown, and I have added a primer which is binding somewhere here, and this is what is happening in the tube which is containing dideoxy GTP. Okay, so the dideoxy GTP tube will have a strand which stops here, and when it stops here, I'll get a piece which is the shortest, which will be in that tube where I added dideoxy GTP. But this tube will also contain 2 more pieces. Okay, and what are those pieces? Those pieces are where dideoxy GTP was added here and dideoxy GTP was added here. Okay, now see what happens in the piece where dideoxy ATP was there. Now there I get a piece which is 2 length, after which it stops. So that is there. Then I'll get another piece which is length of 1, 2, 3, 4, 5, 6 and 7, which is here 6 and 7. Likewise, in the tube which contain dideoxy CTP, I'll get a piece which is of length 3, and then a piece which is of length 1, 2, 3, 4, 5, 6, 7, 8 and 9. Okay. And the tube which has dideoxy TTP, I'll get a piece which is length of 4 and a piece which is length of 10. 3, 6, 9 and 10. Now see the beauty, in every tube the piece you will have will be of length which is not present in any other tube. So now all I have to do is look at which tube ka sample I loaded in with gel. So remember, this is where I added dideoxy ATP. This is the tube where I added dideoxy CTP. This is the tube where I added dideoxy GTP. This is the tube where I added dideoxy TTP. And I do autoradiography and I can directly read the sequence. See, G, A, C, T, G. That is G, A, C, T and G. Correct? And then further A, G, C, T. A, G, C and T. Now, this is the tricky part now. Once I know this sequence, I have to realize that my DNA strand whose sequence I am going to try and find out will be complementary to this. Okay. So see, once I do this, then I have to go back and do the opposite. Where I read C, it is actually supposed to be a C. Where I read A, it is supposed to be a T. So I read the sequence correctly now as A, G, C. See, A, G, C, T, T. A, G, C, T, T, T. And then C, A, G, T, C. C, A, G, T, C. And this is exactly complementary to this sequence. Okay. So, everyone got the hang of it? Okay.
Yes, sir.
Now,
Yes,
as
sir.
somebody said, okay, here also I'm kind of reading manually. Okay, the issue is can I do it automatically? But before I get to that, there is another slide I want to share. For this work, Gilbert and Sanger, for their work in 1977, got the Nobel Prize for Chemistry in 1980. Just 3.5 years after they invented this, people realize how important that is. So in that year, ironically, the Nobel Prize was shared by 3 people. He got 1/4 of the money, he got 1/4 of the money, and Paul Berg got half the money. And what did Paul Berg do? He discovered all the fundamentals of DNA, rDNA. Remember that cut and paste technology, restriction enzymes and all that we talked about? That was all partly Berg's inventions or discoveries. So essentially in a sense, for recombinant DNA technology and biochemistry is what these prizes were given under the heading Nobel Prize in Chemistry. Okay? Now, see what I do if I want to automate. Okay, if I want to automate, what I do is I use what are called as fluorescent dyes. Okay. Now, what do I mean when I say a fluorescent dye? It's a dye which will do fluorescence. And what is fluorescence? I shine light of a certain wavelength, and this dye will emit that absorbed light at a different higher wavelength. Okay. And generally, many times, this might be a wavelength where you can see color. Okay. For example, I shine UV light and it will emit, let us say, yellow color, or I shine UV light and it will emit red color, and so on and so forth. Okay. Now, there are 2 ways I can use these fluorescent dyes for automation. Okay. I can do one thing. I can use this fluorescent tag to tag one end of the DNA. Okay. Just like we did P32 labeling, no? I can tag the primer with it. Okay. So which means I'm going to tag the 5' end. And therefore, every piece that I generate, because it always contains the primer part. Remember, when I showed here, the primer is always present in every piece. And let us say I've tagged it here, then every piece will have that fluorescent tag. Okay. Now, the other way I can do is I can tag actually dideoxy analog. Okay. And I can tag different dideoxy analogs with different fluorescent dyes. Okay. Which means each of this dideoxy analog, when you shine light on it, will fluoresce at a different wavelength with a different color. Okay. Now, for automation, generally what they do is they use what is called as dye terminator sequencing. That means what? The terminating nucleotide, which is dideoxyadenine or dideoxyguanine or dideoxythymine or dideoxycytidine, they are the ones which are tagged. Okay. So now see what will happen when I do that. When I use a different color tag for each of the different dideoxy analogs, when I run the gel, what I will find is the different gels here, whether this was a guanine, dideoxy guanine wala gel, this is a dideoxy adenine wala gel, they'll all have different colors. For example, let us say this will have green color. Then this one here, which is a thymine, will have red color. This will have blue color, and let us say A will have green color. So now what I can do, instead of reading it manually, what I do is mix all the samples and run it in one shot. Then I know that when green comes out, it is adenine. When black comes out, it is guanine. When red comes out, it is thymine. When blue comes out, it is cytosine. When red comes out, it is thymine. When black comes out, it is guanine. So essentially now what will happen? I can run it through the gel, a single gel, and I just wait for the thing to come out of the gel and monitor which color is coming. Okay, and see what I do. Depending on what color is coming, I can directly read now. Okay, so green is A, then black is G, red is T, then blue is C, then again red comes, it is C, and again black comes, it is G, and so on and so forth. This makes my life a lot easier. So I can now automate. Okay, and see what I do when I automate. Here is an example. I've shown it in color. Okay, so dideoxy T is red, dideoxy A is blue, dideoxy G is green, and dideoxy, okay, I should say purple and blue. Okay. Now, in the different test tubes, when I generate different size of pieces, they'll all end with different colors. This will all end with red color. This will all have an end which is purple. This will all have ends which are green. Then I mix them all and I run it in one shot and I just see which color is coming out. And depending on which color is coming out, I can directly read the sequence. Okay, and this is how I can do automation. Now I don't even need a person coming there. The machine will do on its own. Okay, and I'll get the sequence. Even it will also read out the sequence and give it to me, because all you have to do is program and say, when you see signal at this wavelength, mark it as T. When you see signal at this wavelength, mark it as A, and so on and so forth. Okay, so nowadays all this is done automatically. Okay, so what we call a Sanger auto sequencing. Just to give you a cost of it. Okay, if I wanted to sequence a human genome, no, using Sanger method and using this auto sequencers, I would need hundreds of scientists. I would need hundreds of machines. It will cost me about $3 billion, and it might take me 10 years. Okay. But just to give you a feel, if I apply what are called a second generation and this new generation, third generation sequencing, see what has happened. Suddenly I need one or two scientists. I can do everything on a single machine, and the cost is about $1,000, and I can do it in an hour. So it has become that fast nowadays. Okay. So to Maxim Gill, take DNA, tag it at the end with 5 prime, at 5 prime radioactive phosphorus, then cut at different positions using either DMS formic acid, hydrazine or hydrazine sodium chloride, and then run all those separate different pieces on a gel, and depending on the size of the piece, you can read the sequence. And in Sanger dideoxy, what you do is the unknown pieces actually copied using a known primer and DNA polymerase, but you do so under different conditions. One where you have dideoxy ATP present, in the other case dideoxy GTP, and the third case dideoxy CTP, and so on and so forth. And after you get all these different sizes of pieces, because they have been chain terminated, you run it on a gel, and again, depending on the piece, get the sequence and then do the complementary of it to give you the real sequence. Okay, so this is a comparison of the two. Again, Maxim Gilbert is based on chemical modification. This is based on termination using dideoxy nucleotides. Developed by Maxim and Gilbert, developed by Sanger and his colleagues. Chemical method, enzymatic method. Uses a lot of hazardous chemicals and radioactive material. less hazardous chemicals. It's very sensitive and very specific. This is slightly less sensitive, and sometimes it will do errors, okay, because DNA polymerase sometimes makes mistakes, okay. So sometimes you have to reread. So a lot of people, if they have an unknown piece of DNA, will do the sequencing experiment twice or thrice to make sure the results are, what you call as consistent before they will stop, okay. Now, if you have 5 minutes, I can give you a glimpse of what is called as next generation sequencing. One method of that is called as pyro sequencing. Okay, this was invented in 1996, almost 20 years after Sanger and Gilbert method, and see what it does. Okay, uh, it's a very interesting technique. It looks at the release of pyrophosphate when polymerization occurs. Okay, remember I showed you when dideoxy or deoxy NTP is added, the polymerization releases pyrophosphate, and this pyrophosphate is the one which is detected in this pyrosequencing. But see how it is detected. Very interesting. Okay, now uh, here is an example. I have a polymerization reaction that has occurred, and when polymerization reaction occurs, what happens is pyrophosphate is released, right? And this pyrophosphate which is released is taken up by an enzyme called ATP sulfurase. Okay, and see what ATP sulfurase does. The substrate for ATP sulfurase is adenosine. This is adenine. This is adenosine 5' phosphosulfate. That is why it is called APS. Adenosine 5' phosphosulfate. Now see what this does. This will take the pyrophosphate which it has got from the reaction where polymerization has occurred and put that pyrophosphate onto APS releasing sulfate. So what it does is it uses the pyrophosphate to reform ATP. Now what is done is that this ATP is used very interestingly to actually catalyze a reaction. Okay, a reaction where luciferin is converted to oxyluciferin by an enzyme called luciferase. Okay, anyone heard of this enzyme luciferase by any chance?
No, sir.
No, sir.
Okay, okay. How many of you here, for vacation or when there is a big holiday, go to your village or small town? Okay. In other words, you have relatives in a small town or a village. Okay. When you go there for vacation, no, you find that things are more open. Okay. There are not too many buildings. There are a few houses, and there is a lot of greenery and all. And at night, no, when you sit in the veranda after lunch, after dinner, and you look out at the scenery, you sometimes see this little light which will go on and off. Have you ever noticed that by any chance? गांव में किसी ने भी इसको firefly बोलते हैं. Okay, fireflies.
Yes, sir.
Okay. Ah,
Yes,
and
sir.
sometimes no, when you're little, no, you go and catch these fellows and put it in a little bottle, and then when you cap it at night and look, they'll, when they're flying, they'll give out this little light. Okay, and this light that they give out is because they have this enzyme called luciferase, and this luciferase in the fruit fly, or I should say not fruit fly, firefly, converts luciferin, which is produced by the fly itself, to oxyluciferin, and when this reaction occurs, it generates a little light. Okay, and you see that light as it comes out. Okay, now see, this principle is used in the sequencing. Now see what happens. I'm trying to do a sequencing, right? And when I do sequencing, uh, let us say I add one of the four bases. Okay, let us assume for example that yellow is ATP and blue is say GTP, just for argument. Okay, so I'm sequencing this space. I've added a primer, then I put ATP, and when I put ATP, I find nothing happens. ATP doesn't get added here because it is not complementary to the green. Okay, what is complementary to the green is blue. So when I add ATP, no reaction occurs. When no reaction occurs, there is no pyrophosphate. When there is no pyrophosphate, there is no ATP. When there is no ATP, there is no light. Now what I do is, take out, wash away all the ATP and add GTP. The minute I add GTP, what happens? GTP is recognized as complementary to the green. It comes here and this polymerase joins together this G to this strand, and when it does so, it releases pyrophosphate. And when it releases pyrophosphate, immediately I get ATP formation and I see little light. So if I add GTP and I see light, I say, oh, at that position there is a G. Okay. Now the same thing will happen that when I add CTP and TTP, I will not see any light. Okay. Now again I repeat the cycle. Now blue has already been added, right? So now in the next step, after blue has been added, look at what is there. There is a green. Okay. There is another green. So again when I add ATP, nothing will happen. Again when I add GTP, I will say light. Okay, in fact, what will happen is when I add GTP, because there are two greens and there are two blues here, I will generate more pyrophosphate because two will be added and I'll get more light. Sometimes when I get more light, then I know there were two bases which are added. When I get 3 times more light, there were 3 bases. Okay, so essentially what I do is every time I see a pulse of light and that light is correlated to which base was added, I will know what the sequence is. Okay, now as you realize, if there are a lot of greens in a row, and because of that, as soon as I add GTP, a lot of blues are added. After a while, there is too much pyrophosphate, and I might not know whether there is 5 release or 6 release. So one of the biggest disadvantages of this pyrophosphate sequencing is if there are too many same bases, one after the other, it will make an error. But other than that, if there are 1 or 2, 3 at the most, then this works very fast. Okay, and that's why it is called as a next generation sequencing. It was developed by a company called 454 Life Sciences, and therefore the method is called as 454 pyro sequencing. Okay, but later because of this issue of errors, okay, when there is a lot of same bases, this method was actually discontinued. Okay, now it is used but used very sparingly, but there are a lot of other methods which are used now, like Illumina sequencing, solid sequencing, ion torrent sequencing, single molecule sequencing. And the thing is, all of them are amenable to automation. And it is these methods which allow you to actually sequence an entire genome with 1 or 2 people working on 1 or 2 machines in a day or 2. Okay, and it has made sequencing very routine. At one time, sequencing itself used to be a big experiment that you did, but now it is something which people do on the side. Like अपना chemistry lab में अगर melting point को automate कर दिया तो कितना अच्छा पड़ेगा ना वैसा. Okay, so nowadays it is like Amazon. You want anything, you can get it on Amazon. Whereas if you want to sequence, you just do one of these and it's afternoon's job. Okay, but for sake of realizing where we have reached, it is very important that you know these 2 methods, because they tell you a lot of basics of chemistry, basics of biology, basics of genetics, and all these information was used to design how to sequence DNA. At that time, it actually revolutionized everything. That's why they got a Nobel Prize. Okay, many of these techniques are actually a fallout of some of the knowledge gained by these 2 methods. Okay, so in a sense, many times they are not given as much importance as the first method, which was Maxim and Gilbert. Okay, so I'm going to stop here for the day. If anybody has any questions, feel free. I'll do the stop sharing and come back to my actual regular screen, and let us see anything in the chat here. Nothing in the chat, but last time who had questions? If we add alkaline phosphatase first, radioactivity first, and then alkaline condition to separate DNA. No. What we do is you add NaOH to separate the 2 strands. Then after the 2 strands are separated, when you run it on the gel, okay, you make sure that you do not neutralize them, because if you do, they'll all come together. But remember, the gel is run in the presence of SDS, which is a detergent. Okay, and in that NaOH doesn't do anything to that gel. Okay, and then after separation, you actually neutralize. And after you neutralize, then you actually uh, again add a little bit of buffer, which is basic, and then add phosphatase. Okay, and that removes the end phosphate, and then again you make it neutral, and then add your kinase, and then you put that tag of P32. Okay, in both methods we talked about controlling amount of enzymes. Okay, Aditika question. Uh, to get cuts at different places on the sequence. How exactly do we calculate it? It will differ for different lengths. So how exactly do we calculate it? Okay. Actually, initially when the method was new, this used to be a big exercise. Okay. Now what you find is these methods are so well known now that when you want to do the sequencing, if you read the appropriate reference, for example, Maniatis, or even if you go to the website of the people who sell these reagents, it is almost like cookbook. Okay, okay, take this many microgram per DNA. When you take this many microgram per DNA, you will realize that it will only contain so many bases. Okay, doesn't matter what the length is. Okay, and then because of so many bases, 1/4 of that will probably be adenine. Okay, and if 1/4 of that is adenine, how much of your reagent should you add such that you will not cut everywhere there is adenine. So let us say the number of moles of adenine is 1, then you end up adding 0.25 moles of this reagent, so that it never actually will able to add cut everywhere. Okay, so that is how we do this calculation. Okay, but in Maxim Gilbert initial days when it was being developed, this used to be the most tricky thing. Okay, and therefore some labs used to do Maxim Gilbert without a problem because they had this protocol down very well. Some labs used to struggle for a while, but Sanger deoxy is much easier. These methods are now routine. Okay, so if you just look at any book on life sciences or mol bio, they'll actually tell you recipe for doing Sanger dideoxy, how much of each to add. Okay, so even though lengths are different and bases are different, in general we use the probability of any base occurring 1/4 of the time for doing the calculation. Okay, does that answer your question, Aditi?
Yes, sir.
Okay, so any other question? Any other question? So shall we stop for the day then?
Yes, sir.
See, okay, so I'm going to stop recording.